A little preface, but we have a cat in studio today. If you hear some random noises, some random chains, some meowing, purring, I apologize in advance. Hello everyone, and welcome to episode 3 of the Asclepius podcast, where we talk all things medicine and science with your host, Timothy Jew. Now, today we have our very first special episode, and not gonna lie, I've been dreading this episode for quite some time. I've literally finished writing the scripts, editing, mastering, making the music for, and rendering every other episode for the season before this one. And why, you might ask? Well, my fellow learners, it's because I thought that filling an hour of time with my own ramblings and ideas seemed extremely indigestible, and it also seemed like the ultimate exercise of vanity and self-importance. Also, let's just be real here. I'm not the most eloquent speaker, and an hour of time just seems like a lot of potential for mistakes and hot takes. Anyway, the solution that I've come up with is to make these special episodes like 15 minutes long so you don't have to suffer through them for too long, and so I can reduce the time I expose my IQ, or lack thereof. Also, just a quick Timmy tangent. I have no idea what my IQ is, and although I don't really care how intelligent I am, is it weird that I sort of don't want to ever take an IQ test? Like, what if you do know where you place on the bell curve of this acute definition of intelligence? I mean, it feels like you're just further reducing yourself into bits and data and slowly stripping away your sense of agency and identity. Or are we but little marbles floating around in a vast... Okay. That aside, for this podcast, well, it's just me talking about pediatric lung transplants, doing a quick review on cystic fibrosis, researching flow cytometry, fluid dynamics, trying to figure out why pediatric lung transplants are something to marvel at, and so much more. Now, I'm going to heavily preface this episode by stating that I am not a physician or even a medical student at this point, so all this information is simply my interpretation of research and data that I happen to look over in response to reading some surgical news. So think of me as your little science journalist for today. Anyway, please don't forget to like, comment, rate, subscribe, share, and all that good stuff. It really helps the algorithm and will get the information out quicker. Regardless, hope you all enjoy. Oh, hi there. So before we start today's episode, let's go over our sponsor, us. Now, if you are dying to part with your hard-earned cash, then feel free to support us on patreon.com/the Asclepius podcast. That's TheAsclepius podcast. Perks include being able to ask questions to future guests, suggesting future guests, getting cool original merchandise, and so much more. Hopefully, this is the first season, so we don't know how it's all going to play out, but either way. And once again, visit our patreon.com/the Asclepius podcast, or visit us at asclepiuspodcast.wixsite.com/home. Eventually, we'll try to get our own domain too. Anyway, look forward to seeing you soon. Are you extremely gifted? Do you have complete mastery over the arts? Well, my friends, if you are literally Leonardo da Vinci, DiCaprio, reincarnated, I would highly encourage you to participate in the seasonal Asclepius Podcast Art Challenge, where we have but the most able of artistic maestros submit their rendition of the Rod of Asclepius. Those that happen to edge out their competition will have their art not only used as our logo for the season, but will also have their art used as merchandise, with all the proceeds of that merchandise donated to Children's Hospital. How altruistic of you. If you are interested in showcasing your amazing talents or just helping out Children's Hospital or the podcast, then feel free to submit your art at our website at asclepiuspodcast.wixsite.com/home. Anyway, let's get back to the episode. Now, this episode is going to take your breath away. But don't worry, people, because it's also going to give it back. We're talking about lung transplants. Crazy amazing stuff. Now, I've always viewed transplants as almost Shakespearean, a multi-dimensional conflict laden with controversy and drama. But instead of ending with a whole lot of death, par for the course with Shakespearean stories, we have a more optimistic and amazing prospect of giving life back to those that never had a chance in the first place. The classic story of anabolism and catabolism. Today we will go over the science and mechanisms that contribute to such a feat, but also we're going to touch on one of my favorite little foods for thought, bioethics. So let's get into it. The NYU Langone Transplant Institute was able to perform their first pediatric lung transplant on 16-year-old Alejandra, who was suffering from cystic fibrosis, on November 1st, 2021. The operation was performed by the director of NYU Langone's lung transplant program, Dr. Stephanie H. Chang, and team. What a dope achievement, right? Anyway, upon reading this great news, I couldn't help but wonder why pediatric solid organ transplants would be any different from adult organ transplants, and why lung transplants in particular are viewed as so much more difficult. So, in the spirit of good science, of course I made my own hypotheses before starting this venture so I could test these out. One aspect I definitely thought of first was the potential social context and legislation. All of this just feels like your traditional muddy ethics which are at play with conventional transplantation, pig heart transplants, I'm looking at you, except all heightened now due to the fact that we are dealing with children here. Another prediction of mine was the extreme variation of donor and recipient organ sizes, as well as the potential fragility of said organs, particularly the lungs. And obviously the last potential factor I could see was that the smaller structures would just generally be harder to manage, but that factor seems like the most underwhelming answer that perhaps at this moment I have difficulty appreciating. Anyway, another reason why this question was plaguing my mind so much is because of my conception or misconception of transplantation morbidity and immune response. Now, hear me out. I know this is wrong, or partially wrong, but my original assumption was that children would be more likely to have less immunological responses to foreign tissue since they would not have as much time to develop antibodies and mount a response, right? Well, as it happens, I had a recent conversation with a cardiologist and he basically just said, nope, it's way more complicated than that, buddy. So, anyway, with my questions and hypotheses set in place to guide my Google searching, let's explore the crazy world that is pediatric lung transplants. Also, before we start diving into the metrics and the logistics, let's not forget Alejandra for being so brave and accepting to go through this procedure that would change her life, the physicians and healthcare workers that participated in this amazing feat, and of course, to the donor who allowed the end of their life to be the start of another's. So, before we start getting into pediatric lung transplantation, let's get into why Alejandra needed this procedure to be done in the first place. Cystic fibrosis. Now, from what I remember in my college Foundations of Biochemistry course, cystic fibrosis and all of its problems originates from mutations of the CFTR gene, or the cystic fibrosis transmembrane conductance regulator. Now, do you all hear that? If you think that's a record player playing a broken record, then you'd be correct, because that is the broken record of biology and physiology, playing over and over again. Just like we've talked about in our first episode, the topic of balance is at the center of all of this. Whether it be Le Chatelier's principle, osmosis, or what have you, entropy loves winning, and spoiler warning, it often does. In this particular instance, we have entropy applied in the context of water following salt across membranes to achieve osmolarity balance. We like having our ion concentration, that is, the amount of ions per volume of water, to be equal across our membranes. So the normal function of the CFT regulator is to regulate the flow of sodium to our mucus. This creates imbalance in our ion concentration because now we have a buildup of ions on one side without any water to accompany these lonely ions. This is where entropy comes into play, where our water will start moving through the membrane to follow the movement of ions into our mucus, ultimately resulting in our mucus being nice, light, and hydrated. Now, mutations of the CFTR gene, as you can imagine, can lead to no movement of sodium into the mucus, which then leads to no movement of water across the membrane, and we end up with this thick, dehydrated mucus, which can have drastic and dangerous effects. For instance, having thick mucus coat the alveoli of our lungs, the little balloons which are the site of carbon dioxide and oxygen exchange, can actually create a greater external pressure on those balloons until they eventually cave in on themselves, leading to a lack of oxygenation. And oxygen, as we all know, is at the center of thousands of metabolic pathways that we need in order to stay on this side of the fence. This issue is so dangerous and common, in fact, that half of the babies that were born in 2019 with cystic fibrosis are expected to live past 48 based on the 2019 registry data. Half get to live past 48. So, with all this information in review in our heads, one may find themselves questioning what we should and could do about this. Let's finally start talking about pediatric lung transplantation, shall we? Upon reading long-term outcomes of children after solid-state organ transplantation by John Jin Kim and Stephen D. Marks, we see a lot of my original questions and hypotheses answered, validated, and questioned, which is all great news. The paper starts by talking about what insane hurdles we had to overcome for pediatric solid organ transplantation. The first set of improvements that we made was due to better surgical techniques with smaller structures, which of course makes sense, but let's be real here, that's sort of underwhelming. I mean, don't get me wrong, I love the thought of improving your technique with further specialized instrumentation to save lives, but from an academic and learning the science standpoint, it doesn't really tickle my fancy might tickle. Unsurprisingly, the greatest area of improvement was based on breakthroughs with immunity studies and screening developments. Predominantly, what seems to be a huge deal in the world of transplantation right now is the ability to accurately identify these little things called human leukocyte antibodies, or HLAs. Now, before we get into how we screen for these little things, let's do a little review on HLAs, or in more potentially familiar terms, the major histocompatibility complex. Essentially, one of the processes that allows a cell to maintain homeostasis is to identify foreign substances that the cell can encounter and then destroy them. Turns out, after generations of statistical shaping, evolution has found it pretty nifty to absolutely obliterate foreign substances from the body. We have class 1 and class 2 major histocompatibility complexes, or MHCs. Human leukocyte antigen A, B, and C are class 1, and human leukocyte antigen D is class 2. There is a class 3, and there are some more specifications, but that might be a little too tangential for today's lesson. The way that I remembered it is by asking myself, who is number 1? That's right, me. So, class 1 MHC molecules are going to identify the peptide fragments that are produced within your own cell. Who made it? Me. And I am number 1. To get a little granular, we have these proteasomes which degrade proteins by cleaving peptide bonds, and some of these fragments, usually 8 to 10 amino acids long, can actually interface with a cleft or binding site of the HLA, which will then be shipped through the Golgi apparatus and eventually find its way to the surface of the cell. This will then be presented to the CD8 positive T cell, known as the cytotoxic T cell, to get abolished. CD8 is a protein that interfaces with the class 1 MHCs, just to let you know. So the type of protein that the T cell contains will actually determine what type of T cell it is. Anyway, this process was really cool to me because I remember reading in my Kaplan MCAT book about how certain viral vectors avoid this pathway once infecting a cell by no longer creating MHC1 molecules. This makes sense because viral vectors often utilize the machinery of their host cell to make the proteins that they require to wreak havoc, and since cells that create the MHC1 molecule tend to get killed, the virus would find some mechanism to avoid that. However, despite the virus's best attempts, we still have a rebuttal to this ingenious strategy, which is the natural killer cell, who's like, hold up, where's your password, fam? And then just whoosh, swiftly dispatches the infected cell. This could also be present in cancer cells, of course, because once again, this is class 1, and that means you made it. It really be your own cells sometimes. So, what about class 2? Well, we have professional antigen-presenting cells such as dendritic cells, macrophages, and B cells, B standing for bone marrow. And these are essentially going to gobble up some foreign proteins via endocytosis and then present these peptide antigens on their surface. The major histocompatibility complex 2 actually has a site specific to the CD4 protein as opposed to the CD8, which is attached to the CD4 positive T cell also known as helper T cells. These helper T cells will then bind and do a number of things to assist and make the whole process faster, whether it be releasing cytokines, cyto referring to cell, and kines referring to movement. Helper cells can actually activate/differentiate B cells into either effector cells, which are going to produce a bunch of antigens that can bind to the cell and mark them for killing, or they can turn the B cells into memory cells, which will hold that information for future instances so that it can mount a faster attack. In a normal scenario, your body will create its own unique set of peptides in its own way, which will be comfortable to its own system. You might be wondering, Tim, what's the difference between a tyrosine from your body versus a tyrosine from another body, or from a bacteria or a virus? What do you mean by a foreign peptide? Well, my dear listeners, I've scoured the internet for this answer and I have not been able to find anything. It's been 3 days of searching and I think it's finally time to just let it go. I even asked the doctor I was working for about this very topic and he said, in general, dude, I've taken immunology like 14 years ago. You're closer to the answer than I am. Lol. Google it. So, my open-ended, currently unanswered hypothesis is that it might not be the individual tyrosine molecules themselves, but perhaps the unique sequence of 8 to 10 amino acids that is produced from foreign sources that would not originate from our own genetic source code. Okay, breaking in, this is Tim from the future of writing the script. So I recently just had a discussion with another doctor about this topic and they actually said that I was correct. So I guess it's not really the amino acids themselves, but their short sequences. Nice. Anyway, going back to the topic of our cells making their own peptides, if a cell of yours has been degraded via apoptosis or what have you, its contents will float around and, assuming you don't have an autoimmune disorder or anything along those lines, your body will just dispose of the material in a normal fashion. If there are foreign protein fragments floating around, your body will dispose of them in a horrifying immune self-defense thing. And this is the problem with transplantations, because our organs are producing our own peptide fragments, and when introducing our fragments to a new biological institution, such as the recipient of our organs, it will be recognized and then destroyed. Our unintelligent little components unknowingly compromising the larger biological system. Quick tangent. This reminds me of a book I read called Gödel Escher Bach, An Eternal Golden Braid, where Douglas R. Hofstadter, the author, refers to the lower level hardware of the brain, i.e. neurons in the various cells that comprise the brain, to that of individual ants. He then goes on and relates the brain with its complexity and dynamics to the complexity and emergence of organized pattern that we can observe within an anthill. Similar to these ants, our cells that are so integral to our immune system are not intellectually competent, at least by our widely accepted definition of intelligence. Our immune cells can't just step back from their menial tasks of releasing cytokines or interfacing with certain antigens, no more than an ant can take a step back and realize that it is following pheromones and wondering why it is grabbing a leaf through this particular trail. And sometimes this can be at the dismay of the overall system. Sometimes sacrifices to these individual lower level constituents can actually contribute to the sustenance of the overall system, such as the case with ants being eaten by anteaters to keep the anthill safe, or such as the case with damaged cells that undergo programmed apoptosis to ensure a cohesive biological system known as us. I'm sorry for the tangent. I just thought that this topic was too cool to not talk about, and perhaps I'll go over this topic in another special episode because I just really can't get enough of this book. Chef's kiss. Mwah. Getting back to the topic at hand, our bodies lack the insight to appreciate foreign invaders when it comes to transplants, and this is where there is a need to recognize the types of molecules and antigens we are dealing with so that we can preemptively screen for compatible donors and recipients. Now, the way that we classify these cells is through this little nifty thing called flow cytometry. It's actually so interesting, but I will avoid going over everything that I've learned about this particular concept because I'm currently 7 research papers in, and I've even called Beckman Coulter, the company that makes these machines, to address some questions I had with the application of Bernoulli's principle and other fluid dynamics used in this machine. Yeah, I go hard, baby. However, I will reduce the absurd, amazing science that comprises this crazy process into its main ideas. Flow cytometry utilizes a process called hydrodynamic focusing, which can isolate single cells into a single string. The way they do this is by having a central fluid containing our samples of interest and a sheath fluid. the sheath fluid goes faster than the central fluid, which supposedly compresses the central fluid in a precise manner that allows for us to get a single stream of single cells. Now, why did I call Beckman-Coulter to discuss the principles of Bernoulli here? Well, because that's not how Bernoulli's principle works from what I've learned. Slower fluids should have a greater pressure on the side, not faster fluids. Anyway, I digress. The takeaway here is that we can literally get single cells lined up after each other. How cool is that? Now, this stream is going to have a bunch of lasers pointing towards it that all eventually bounce their way to some detectors that are hooked up to some algorithm computer craziness. To simplify, there are two types of lasers that I will be talking about. Ones that shoot inside the cell to see how complex it is, and one that shoots around the cell to see how large it is. For the laser that shoots into the cell, we get greater diffraction patterns the more complex the cell, due to more obstacles that interact with the laser beam. The other laser works by realizing that larger cells occlude more light reaching the sensor across the laser. Once we get the size and the complexity of the cell, we can then classify the types of cells that we have and be like, dang Cynthia, that makes 12 eosinophils and 4 astrocytes. You then just grab the professional antigen presenting cell or whatever, lyse them, study them, and we're good to go. This is paired with lab work to see which patients are eligible for the operation. In particular, panel reactive antibodies are screened to calculate the probability that the patient's antibodies will attack the organ, leading to rejection. Amazing, right? Another cool thing that helps us with transplantations right now is the fact that infancy is an immune-privileged time due to less developed immune systems. So, oh my goodness, I was sort of correct with my original hypothesis. Now, when I previously told a cardiologist about my hypothesis, I said that children in general have less developed immune systems. So, technically, I'm not 100% correct here, but I was correct in assuming that nascent immune systems might be a little more flexible when it comes to transplantation, even across incompatible blood types. Amazing stuff. Now, let's get into the rest of the hurdles that transplant surgeons have to overcome. First, we have immunosuppression, which is our main tool that we use in tandem with organ transplants, and this turns out to be a common cause of a lot of death, or rather, one of its many side effects can often be the cause of death, because our immune system, in many instances, actually helps us survive. The predominant mechanism of action for these immunosuppressants is to actually interfere with the release of cytokines by those helper T cells or CD4 positive T cells, as we previously talked about. And in general, just anything that would hamper our normal immunological responses would work. The current aim is to minimize side effects while maximizing the benefits. Also, to better understand potential markers that indicate a statistically safe time to wean off the immunosuppressants. Another crazy thing that we need to talk about is post-transplant lymphoproliferative disease. Post-transplant means after the operation, of course, and lymphoproliferative disease means that the lymph cells or the lymphocytes will start multiplying too rapidly. Now, why does this happen when on immunosuppressants? Well, a very common disease that we often encounter when we are young is this virus called the Epstein-Barr virus, not that Epstein. Now, for the most part, it remains dormant in our system due to our immune system, so using immunosuppressants can actually reactivate the Epstein-Barr virus, which can then infect our lymphocytes, leading to PTLD. There is also PTLD in Epstein-Barr virus naive patients, or patients that have not been exposed to the Epstein-Barr virus yet, and researchers are currently not entirely sure what is the cause of that. Epstein-Barr virus negative post-transplant lymphoproliferative disorder by Nelson et al. Essentially did what seems like a literature review and found that, as with most things in medicine, it is not that black and white. There are various EBV negative PTLDs that are not affected by decreasing immunosuppressants, leading to a distinct etiology. And those that are affected by decreasing immunosuppressants are somewhere in the middle. Symptoms of PTLD, by the way, include fever, swollen lymph nodes, general malaise, among others. Another big cause of death after pediatric transplants is cardiac failure, where we have increased global arteriosclerotic disease and calcification with increased arrhythmias due to the immunosuppressants. Reading Cardiovascular Toxicities of Immunosuppressive Agents by Miller et al., we see that the main culprit for cardiovascular toxicities is calcineurin inhibitors, which led me to read Calcineurin Inhibitors by Omar et al. To learn about their mechanism of action. Apparently, they work by binding the cytoplasmic receptors known as immunophilins, and the whole drug immunophilin complex competitively inhibits the action of calcineurin. a phosphatase. The inhibition of calcineurin after a couple of steps inhibits the translocation of several transcription factors coding for several cytokine genes such as interleukin, tumor necrosis factor, granulate macrophage colony stimulating factor, and interferon gamma, which all reduces our immune response. Now, how does any of that affect our cardiovascular system, you may ask? It apparently is an unknown area of science, and despite my best attempts at uncovering any potential mechanisms of action, it seems like the numbers are not large enough to fuel a statistically significant study as of yet. In fact, a recent conversation I had with a cardiologist, yes, I harassed the doctors that I'm working with a lot, uncovered the list of potential pathways and ambiguity of whether these cardiovascular effects are resulting from some downstream pathway by affecting interleukin 4, 6, yada yada, or if it is more related to the macroscopic view of the physical state that the patients are exhibiting. So I would like to apologize, my dear listeners, for the fact that I was not able to get a solid answer for you. But I also wanted to just say that this is sort of great news. More science and learning to be had. Anyway, since we observe calcification in arteries after transplant, it might make sense to combat that and reduce the potential for heart attacks and whatnot. So statins are often paired with immunosuppressants to reduce the buildup of cholesterol. And just a little reminder, the main mechanism of action for statins is to inhibit HMG-CoA reductase, which is the rate-limiting step for cholesterol synthesis. Another side effect of calcineurin inhibitors is nephrotoxicity due to glomerular. What? Essentially, your kidneys are starting to hurt because their main functional unit is no longer getting blood flow. Yikes. After that laundry list of side effects and inconveniences/obstacles, one can't help but realize the set of concessions we must make in order to sustain life. Speaking of sustaining life, what is an appropriate definition of life? Is it simply an antithesis of death? Is it more? What does it mean to be living? Well, the final thing this research paper goes over is quality of life studies, which I think is so cool and yet so difficult to define. In a later podcast episode, episode 4, we will go over my pseudo-philosophical take on what a physician's role is in society. But until then, I just wanted to finish up this episode by reflecting on the amazing role medicine and science can have in society. Although it's easy to be overcome with cynicism, especially with regards to insurance companies and the overall bastardization of medicine, being able to dissect the patterns that we observe and then translate what we learn from them into tangible products that affect people's lives for the better, whether it be duration or quality, is something that makes me excited to pursue this career. And although perhaps I'm somewhat transiently attached to this crazy world and culture that is medicine, and although I'm only scratching the surface of its imperfections and flaws, I find the perspective of working with like-minded people who have dedicated their lives to help other people such an awesome thing. With that being said, that makes this final episode done. In conclusion, we learned about pediatric lung transplants, we learned about flow cytometry, we learned about human leukocyte antigens, we learned about the difficulties that transplant surgeons have to undergo in order to save lives and to pioneer this relatively new branch of science. Thank you all for tuning in. Hopefully we didn't expose too much of my IQ in the process. If you enjoyed the podcast, hit the like button, comment, subscribe, rate. It really helps. If you didn't enjoy the podcast or you just feel like some constructive feedback is warranted, please also comment below. Anyway, this is the Asclepius podcast with your host, Timothy Jew. I don't know if I'm going to do those stupid name things at the end. The next episode will be an amazing episode with Professor Love, talking about birth control and blood clots and overall lack of communication in medicine. I'm really excited for that one. Anyway, peace.